All right. चलें जी, let's resume. 1 second, please. Okay. चलें जी, let's resume quickly. All right. चलें जी, so this, alhamdulillah, actually summarizes the definitions and the explanations of all of the 3 very important, excuse me, processes, which are active transport, diffusion, and osmosis. Now, the next thing that basically we have to look at is the behavior of animal and plant cells in different solutions, okay? So let's quickly look at that before we, inshallah, I hope we have time for going through the exam style questions. Let's see. Okay. So the next thing, Bacho, that we are going to talk about is, let's talk about, actually, I'm going to highlight it. Yeah, we're 10 seconds, please. Let me go back to my green ink that I love. Okay. So now, again, what we're going to talk about is actually extension of what osmosis is going to be. So osmosis in plant cells. Okay. Again, this is a very important topic from the exam perspective. So please, let's try and understand this thing very carefully. So imagine we have 2 scenarios. Okay. We have 2 scenarios. The first one is we place a plant cell in a concentrated solution. Concentrated solution. What do I mean when I say this? Let me just explain. So that's scenario A. Okay, and then we have technology use करते हैं जरा. देखिए मेरे पास भी ruler है. अच्छा है. And then we have okay. The second scenario in which you place a plant cell. Okay, you place a plant cell in distilled water, not in just in distilled water. And by the way, when I use the word distilled water, that means pure water. Distilled water. Okay, so let's look at the concentrated thingy first. Okay, now I will draw a diagram first. So for example, if that's a beaker, and that is, it has a concentrated solution, which means what? That it has low water potential. Okay, water की concentration इसमें कम है. That's what it means. Now imagine that I place an animal cell. Of course, the animal cell, the plant cell is not this big. Just for the sake of understanding, what if I place a plant cell here? So let me completely draw what a plant cell would look like. Okay. All right. So in a plant cell, let's assume that the water potential is high water potential. So now we need to understand what will be the consequences of the following. Okay. If we're placing this cell in concentrated solution. So what will happen is, now again, all of this is just the explanation of osmosis. So the first thing that we will write is point number 1, the water potential, excuse me, the water potential inside the cell, the plant cell, is higher as compared to the concentrated solution, as compared to the concentrated solution. Okay. So what happens next is, so water moves. Haji, water किधर जाएगा बच्चों? Will it move into the plant cell, or do you think it will move out of the plant cell?
Outside of the plant cell.
Outside of the plant cell, exactly. That's step number 2. So it moves out of the plant cell, of the cell, but we have to understand out of cell that this is incomplete for us to mention. We have to qualify this down. It's sorry, down the water potential, not it's down the water potential gradient. Okay, which of course means from high to low. Okay, that's obviously understood, but I'm just writing for the sake of clarity. So it moves from a region of, I think I will be again needing my ruler to increase this line slightly. Okay. Achha ji. So down the water potential from high to low through, देखिए बस तीनों चीजें मैं लिख रहा हूँ osmosis वाली, through semi permeable membrane, through the semi-permeable membrane, by which process? By osmosis. Okay. Now, let me highlight all of the important stuff that I've written so that you know that all of this has to be written. So that's the yellow color. So we've identified the water potential gradient. So water moves down the highlighter की thickness ज़्यादा कर लूँ, down the water potential gradient, ठीक है जी, through the semi-permeable membrane by osmosis. Okay. Now, something will happen to the cell, and we need to understand what that something is. Okay. And now, what happens is, you know, if I were to redraw the cell again, so it would actually go from this to cell walls are थोड़ी अजीब बन गई है. So there's a reason why I'm actually redrawing this, because ये वाला concept इतना basic है, but this is so often assessed in the exam. It's really, inshallah, I will show you an exam cell question also. So if initially this was the case, that's the nucleus, that's the vacuole. And once the water leaves, what happens is that the vacuole, the pore vacuole shrinks. And, you know, even the cytoplasm shrinks and it gets detached at different places from the cell wall. It looks like the following. So that's the pore nucleus. So what has happened is that, so if that's the cell wall, and this is now the cell membrane, okay? So you can clearly see that the cytoplasm shrunk. Okay, cell membrane आप देख रहे हैं different places से detached. So there are only few points that remains detached, and the vacuole, it actually shrinks. Vacuole shrinks. Okay, so let me complete these points. So after point number 6, I will write, sorry, after point number 5, I will write that number 6, the sap vacuole reduces in size. The sap vacuole, which is, if you can also write just the word vacuole, vacuole decreases / reduces in size. ठीक है जी? And number 7, cytoplasm shrinks. Okay, and detaches itself away from the cell wall, away from the cell wall at different places, as you can see in the diagram that I have drawn. और इस condition को बच्चों क्या कहते हैं? We say that, come on, who is going to tell me, what do you call this scenario as? We say that now number 8, the plant cell is plasmolized. Okay, so that's an important state that the cell is coming in. अच्छा, आप एक चीज, I want you to just understand one thing. You know, you can clearly see I have drawn this transition from this cell to this cell. The size of the cell wall remains the same, actually remains the same. Why is that the case? Don't you think the cell wall should crumple also? It should decrease in size? What do you think, class? Should it decrease in size? What do you think? Yes or no?
No.
Why? Because it's a rigid structure. Usko kuch nahi hoga. But what follows is that the cell membrane shrinks. I have another question. The region that I am shading as yep, the region that I'm highlighting as yellow, what will that region now contain? So all of this region that I've highlighted as yellow, what do you think will it contain? It will contain this concentrated solution. Can anybody tell me why is that the case? If you tell me, I will be the happiest man on earth, honestly, because I will understand that you've understood. Dekhiye, if you remember when we studied about cell wall back in topic number 1, I said that the cell wall is freely permeable. Okay? So once the cell membrane detaches, the solution is free to enter the cell in the direction that I'm drawing like this. You understand? So since if the cell is in the concentrated solution and if the water leaves, what will enter into this region is actually that concentrated solution. You understand what I'm saying? Who does not understand this point? This is really important. बच्चों, do you understand this point? कि ये जो region है, इस region में क्या चीज fill up हो जाएगी? It's basically going to be the concentrated solution. All right, alhamdulillah, that is good. That is good. Now, we need to understand the opposite side of the picture also, okay? If we place a plant cell, so again, let me redraw quickly. Okay, so इसको थोड़ा बड़ा करते हैं. Okay, I really hope you're taking notes all along. If you're not, I would strongly encourage you do. Okay, so let's say this is distilled water, which means it only contains water. Okay, or iske andar, for example, again, if I add, let's say we add another plant cell, let me draw that for you. Okay, of course, I'm not drawing all of those chloroplasts and stuff. That's obviously understood, but it's just to save time. Okay, so now if I say that we're adding a cell, what will happen? So isme hum water potentials ko zara identify karte hain. Now in the solution, you have high water potential. Okay, why do we have high water potential? Because this is distilled water, okay? And inside the cell, there is low water potential. Okay, inside there is low water potential. Okay, ji. So the first thing that we need to identify, I'm going to write that down, the water potential the water. बच्चों मैं आपको बहुत ज़्यादा बोर तो नहीं कर रहा. I'm doing the spoon feeding of writing and drawing is because this is very important. I hope it's not that boring, or is it? I hope everybody's understanding. Come on, give me a yes so that I be happy.
Yes.
Okay, so the water potential of the cell is lower as compared to the uh, solution, dilute solution. So what do you think will happen? Point number 2. So water, अब water कहाँ move करेगा? Will it move into the cell or out of the cell? It will move into the cell by the process. Okay, sorry, down again. The water potential gradient. I'm just sticking to abbreviations. I hope you understand what that means. Water potential gradient, which is again from high to low, but this time the direction of high to low is of course from the solution into the cell. Okay. Number 4, of course, whenever we are talking about the explanation of osmosis, we don't have to forget that we have to write all of these things through semi-permeable membrane, okay, by the process of osmosis. Again, let me highlight all of this important stuff before I proceed. So again, it's the movement of water molecules. It's down the water potential gradient. Okay, and then of course it is through the semi-permeable membrane by the process of osmosis. Okay. Okay. Again, now we need to understand what will be the consequences in terms of, the size, if you like, of the cell. Okay. So let me redraw that again with a purple color this time. So the cell will now become slightly swelled and turgid. बहुत ज़्यादा उसका size increase नहीं होगा. It's just going to be like this. I mean, because of the cell wall again. So now it will be something like the following. Okay, since the water has now entered. Okay, so let me write down the points first and then I will explain. So I will say that the plant cell swells, the cell swells, but and does not burst. That's important. Does not burst. Okay, why doesn't it burst? Due to tough non-elastic cell wall. Okay, non-elastic cell wall. And then the water, इसके अंदर जब पानी enter हो जाता है बच्चों, it literally exerts pressure on the inner side of the cell wall. This pressure is known as the turgor pressure, which is generated by the water itself on the inner side of the cell. So we say that the water exerts pressure. The water exerts pressure on the inside of the cell wall, on the inside of cell wall. Okay. And this pressure is called turgor pressure. And because of the turgor pressure, we say that the cell becomes turgid. Okay. So do you understand this? Come on, tell me, is everybody good with this idea?
Yes.
Okay, wonderful. चलें जी. So ये मैंने सारी spoon feeding कर दी है. This is something that really, the examiner would want to talk about. Now, quickly, what I will do is I will give you a snapshot of what happens. Let's look at, and inshallah, this is I promise that's the last thing that we're doing. I know it's a lot of patience that's required from you. So inshallah, let me quickly paste this picture for you. on my OneNote so that we can go through this also quickly. But if we look at the same scenario with the red blood cell, of course, things will be slightly different. And let me show you what that will be. So in case of an animal cell, this is what the scenario will be. I just want you to look at the diagram. The rest of the points are virtually the same. Therefore, I'm not rewriting all of them. In case of animal cells, the difference is that there is no cell wall. Okay? So if you place it in a dilute solution, that means that the water will keep on entering until there will be a point where the cell will actually burst. Okay, let's look at the points together. Inshallah, you can take notes of them once I send them to you after the class. So the water potential inside the cell is lower as compared to the dilute solution. So the water enters down the water potential gradient through the semi-permeable membrane by the process of osmosis. The animal cell expands and it bursts. Up clearly, the points are exactly the same. I know the points are exactly the same, which we talked about in the plant cells, except for the fact that this will not become turgid, but it bursts, okay? And then the opposite scenario is placing the animal cell in a concentrated solution, okay? Which means that now the water potential inside the cell is higher, which means that the cell will move out of the cell. Sorry, the water will move out of the cell, okay? Down its water potential gradient through the semi-permeable membrane by the process of osmosis. और इसमें जो cell की situation होती है उसको हम plasmolized नहीं कहते. There's a special word and we say that the cell has become crenated. ठीक है बच्चों? So guess what? That's all what this topic is all about. This actually finishes our topic number 2. आज इंशाल्लाह टाइम नहीं है कि हम पास पेपर्स के क्वेश्चंस पे जाएं. असल में I want to spend some time on some practice questions for this topic. So I hope that I've tried to make all of this as clear as I could, inshallah. So इसके मैं आपको जो class notes हैं वो अभी एक PDF file बना के जल्दी से भेजने लगा हूं so that you can have them, and I would love that you quickly make sure that you're taking notes for all of them. So this is diffusion, osmosis, and active transport. What's the date today? Is it 8th or 9th? Anji, bachcho, kya date ho gayi? Okay, great. It's 8th of December, and it's a Tuesday, and it's 2020. All right, there you go. Chalaji, any questions, any comments, any concerns we have for today? Anything that you didn't understand? Anything? Do you think you knew all of this? Especially want to know, but did you knew all about all of these keywords? Because I have really stressed on the keywords today. So if you didn't, you have to ensure that you take hold of all of these keywords. Okay, मैं class खत्म होने से पहले आपको notes भेज रहा हूँ because उसके बाद मैं भूल जाऊँगा, and that's the last thing I want. So quickly, bachcho, no comments, no questions, no concerns. What chup hain aap log, honestly, you're so obedient class. Honestly, you don't bombard me with questions. Usually students, they do. Okay, so combined diffusion, osmosis, active transport. अच्छा जी बच्चों, एक छोटी सी रिक्वेस्ट मेरी और है आपसे. You know, all of the material that I'm sharing with you, you have to ensure that you keep this as a copyright and a confidential, and try and not just spread all of this material around. It's good that you keep it to yourself. I really hope you understand, because copyrights के फिर बहुत सारे issues हो जाते हैं. ठीक है बच्चों? Is that fine?
Yep.
All right. So if you don't have any questions, then inshallah, tomorrow what I'll do is we will do some exam style questions for topic number 2, and we will also begin in our next half of the lesson topic number 3, inshallah. ठीक है बच्चों? चलें जी, अपना बहुत ख्याल रखें. I'm leaving you 2 minutes early. Inshallah, see you tomorrow same time at 8 p.m. sharp. अपना बहुत ख्याल रखें और दुआ में याद रखें. अस्सलामु अलैकुम व रहमतुल्लाह.